Dear students, welcome to our SWAM course, Genetic Engineering and its recent advancements. I, Dr. Neha Sharma, Assistant Professor in Amity Institute of Biotechnology, Amity University, Madhya Pradesh. In this topic, we will discuss on DNA replication in prokaryotes and its comparison with eukaryotic DNA replication. So, question is, what is DNA replication? As we know that reproduction is a fundamental property of all living systems, the process of reproduction can be observed at several levels. First, organism duplicate by asexual or sexual reproduction. Second, cells duplicate by cellular division. And third, the genetic material, that is DNA, duplicates by DNA replication. In simple words, DNA replication means DNA duplication. It was proposed by Watson and Crick in 1953, just after the proposal of double helical model of DNA. He suggested mechanism for its self-duplication. The two strands of the double helicals are held together by hydrogen bonds between the nitrogenous bases, that is adenine, guanine, cytosine and thymine. Adenine always pairs with thymine with 2 hydrogen bonds and cytosine pairs with guanine with 3 hydrogen bonds. Individually, these hydrogen bonds are weak and rarely broken. Watson and Crick suggested that replication occurred by gradual separation of strands of the double helix. During replication, the double helix unwinds and each of the parental strands serve as a template for the synthesis of a new complementary strands. You can see in the next slide that according to the proposal, each of the daughter duplex should consist of one complete strand inherited from the parental duplex and one complete strand that has been newly synthesized. Replication of this type is said to be semi-conservative mode of replication because each daughter duplex contains one strand from the parent structure and one is new. In the absence of information on the mechanism responsible for replication, two other types of replications had to be considered. In conservative replication, the two original strands would remain together after serving as templates, as would the two newly synthesized strands. As a result, one of the daughter duplex would contain only parental DNA, while the other daughter duplex would contain only newly synthesized DNA. In dispersive replication, the parental strands would be broken into fragments and the new strands would be synthesized in short segments. Then, the old fragments and the new fragments would be joined together form a complete DNA strand. As a result, the daughter duplexes would contain strands that were made up of old and the new DNA. To decide among these three possibilities, it was necessary to distinguish newly synthesized DNA strands from the original DNA strands that serve as template. This was accomplished in studies on bacteria in 1957 by Matthew Michelson and Franklin Stoll of California Institute of Technology, who used heavy isotope of nitrogen, that is N15, and the light nitrogen, that is N14. Isotopes of nitrogen used to distinguish between the parental DNA and the newly synthesized DNA strands. So, it was proved by the Michelson-Stoll experiment in which he did. Cells were grown for many generations in medium containing only heavy nitrogen, that is N15, so that all the nitrogen in their DNA was heavy isotope, as shown by the single band, that is blue in color, when centrifuged in the cesium chloride density gradient. Once the cells had been transferred to a medium containing only light nitrogen, that is N14, and the cellular DNA isolated from one generation equilibrated at the higher position in the density gradient, that is purple in band. Then continuation of the replication for a second generation yielded 2 hybrid DNAs and the 2 light DNAs, that is red in color, confirming semi-conservative mode of replication. Now, next is replication forks and bidirectional replication. Replication begins at a specific site on the bacterial chromosome called the origin. The origin of replication on the E. coli chromosome is a specific sequence called ORI-C, where a number of proteins bind to initiate the process of replication. Once initiated, replication proceeds outward from the origin in both directions, that is bidirectionally. The sites where the pair of replicated segments come together and join the non-replicated DNA are termed replication forks. Each replication fork corresponds to a site where the parental double helix is undergoing strand separation and second, nucleotides are being incorporated into the newly synthesized complementary strands. The two replication forks move in opposite direction until they meet at a point across the circle from the origin, where replication is terminated. Now, the new two replicated duplexes detach from one another and are ultimately directed into two different cells. So, replication in the E. coli DNA molecules can be divided into 3 stages. First is the initiation, second elongation, and third and the last stage is termination. Or each steps, different enzymes are required. First step is initiation. The event reflects information derived from in vitro experiment using purified E. coli protein. Initiation involves recognition of origin by a complex of proteins. Before DNA synthesis begins, the parental strands must be separated and transiently stabilized in the single-stranded state. Then, synthesis of daughter strands can be initiated at the replication fork. The E. coli replication origin, that is the ORI-C, it consists of 245 base pair. It bears DNA sequence elements that are highly conserved among bacterial replication origin. The key sequences of interest here are the 2 series of short repeats, that is 3 repeats of 13 base pair sequence, which is yellow in color, and 4 repeats of 9 base pair sequence, which is blue in color. Both repeats have their own conserved sequences. First protein bind to 9 base pair sequences. You can see in figure, which is in slide number 8 under initiation heading, which is showing 4 repeats of 9 base pairs and 3 repeats of 13 base pairs. In next figure, you can see the binding of different proteins. First, about 20 DNA A protein molecules, each with a bound ATP, bind at the 4-9 base pair repeats. The DNA is unwrapped around this complex. The 3 AT-rich 13 base pair repeats are denatured sequentially. This process requires ATP and the bacterial histone-like protein, that is HU. Hexamer of DNA B or helicase protein or enzyme bind to each strand with the help of DNA C protein. The DNA B helicase activity further unwinds the DNA in preparation for priming and DNA synthesis. So, we can say that a number of proteins used to initiate replication at E. coli origin. First is the DNA A protein, binds to ORI-C region. It is ATP dependent, recognize 9 base pair 4 repeats and opens duplex DNA in a specific site. Next is DNA B protein or helicase. It unwinds the double helix of DNA. DNA C protein, it required for binding of DNA B protein at origin of replication. HU, it is histone-like protein, binds to DNA and stimulates initiation. Next is a primase protein, that is also known as DNA G protein. It synthesizes the RNA primer which have free 3' hydroxyl group at the 3' position. First nucleotide makes 5' to 3' phosphodiester bond through 3' hydroxyl group of primer. Next is the SSB protein or the single stranded binding protein. It stabilizes a single strands of DNA which is coming after the activity of helicase. Next is the RNA polymerase. It facilitates DNA activity. Next enzyme is DNA gyrase or the DNA topoisomerase. Second, it helps to release the strain which is generated by DNA unwinding. Next is DNA methylase. It methylated the 5' GATC sequence at Orice C. Several sites that are methylated by the DAM methylase are present in the E. coli origin, including those are 13 mer binding sites for DNA A protein. Next enzyme is DNA ligase. It involves in joining of two fragments of DNA. DNA ligase catalyzes the formation of phosphodiester bond between a 3' hydroxyl group at the end of one DNA strand and a 5' phosphate group at the end of another strand, that is present between two Okazaki fragment, which is present on lagging strand. Now next enzyme is DNA polymerase first, second and third, which is involved in synthesis of phosphodiester bond. So the next question is, what is phosphodiester bond and how it is formed? So DNA phosphodiester bond link successive nucleotides in nucleic acid. All nucleotides consist of nitrogenous base, that is purine or pyrimidine, a pentose sugar and one or more phosphate group. Nucleic acids are polymers of nucleotides joined together by phosphodiester linkages between the 5' hydroxyl group of one pentose and 3' hydroxyl group of the next. The successive nucleotides of both DNA and RNA are covalently linked together phosphate group bridges in which the 5' phosphate group of one nucleotide unite is joined to the 3' hydroxyl group of the next nucleotide, creating a phosphodiester linkage or bond. Thus, the covalent backbone of nucleic acid consists of alternating phosphate and pentose residues, and the nitrogenous bases may be regarded as side group joined to the backbone at regular intervals. Next is types of DNA polymerase. The search for an enzyme that could synthesize DNA began in 1955. Work by author Kornberg and others led to the purification and characterization of DNA polymerase. First, from E. coli cells, a single polypeptide enzymes are now called DNA polymerase first. Other DNA polymerase present in E. coli is called as DNA polymerase second and DNA polymerase third. It discovered in early 1970s. DNA polymerase second is an enzyme involved in one type of DNA repair. DNA polymerase third is the principal replication enzyme in E. coli, whereas DNA polymerase fourth and fifth it identified in 1999 and are involved in an unusual form of DNA repair. DNA polymerase first have following function. It have proofreading activity, that is 3' to 5' exonuclease activity, DNA repairing activity, 5' to 3' exonuclease activity, and polymerization rate is 16 to 20 nucleotide per second, and the processivity is 3 to 200 nucleotides added before polymerase dissociates. Same for the DNA polymerase second, functions are, first they have 3' to 5' exonuclease activity for proofreading. It is functional when DNA polymerase first and third are not functional. DNA repairing activity is also present and polymerization rate is 40 nucleotide per second, and the processivity is 1500 nucleotides added before polymerase dissociates. The last enzyme, that is the DNA polymerase third, it have 3' to 5' exonuclease activity for proofreading, having highest polymerization rate, that is 250 to 1000 nucleotides per second, responsible for elongation process, and the processivity is 5 lakh nucleotides added before polymerase dissociates. We can now relate the subunit structure of E. coli DNA polymerase third to the activities required for the DNA synthesis and propose a model for its action. The holoenzyme is a complex of 900 kilodalton that contain 10 protein organized into 4 groups of subcomplex. There are 2 copies of catalytic core. Each catalytic core contains a subunit for the DNA polymerase activity, that is epsilon subunit, having 3' to 5' proofreading exonuclease activity, and the theta subunit, it stimulates exonuclease activity. There are 2 copies of dimerizing subunit tau which link the 2 catalytic across together. There are 2 copies of processivity subunit beta which are responsible for holding catalytic cores onto their template strands. The gamma complex is a group of 5 protein. The clamp loader, that is gamma, that places the processivity subunit on DNA. Now the next question is, what is the proofreading activity or what is fidelity? An error correction by the 3' to 5' exonuclease activity of DNA polymerase first is known as proofreading activity of DNA polymerase. The function of DNA polymerase is not quite perfect, with the enzyme making about 1 mistake for every billion base pairs copied. Error correction is a property of some but not all DNA polymerase. This process corrects mistakes in newly synthesized DNA. When an incorrect base pair is recognized, DNA polymerase moves backward by 1 base pair of DNA. The 3' to 5' exonuclease activity of enzyme allows the incorrect base pair to be excised. This activity is known as proofreading activity. Following base excision, the polymerase can reinsert the correct base and replication can continue forward. This preserves the integrity of the original DNA strand that is passed on to the daughter cells or strand. This diagram is showing 3' to 5' exonuclease activity of DNA polymerase first. Together, these processes reduce the overall observed error rate about 10 to the power minus 9. Next term is fidelity. It is very important in case of DNA replication. Mismatches in DNA base pairing can potentially result in dysfunctional proteins and could lead to cancer. Many DNA polymerase contain an exonuclease domain which acts in detecting base pair mismatches and further performs in the removal of the incorrect nucleotide to be replaced by the correct one. Thus, the fidelity of DNA replication can be traced to 3 distinct activities. First, accurate selection of the nucleotide. Second, immediate proofreading. And third is post-replicative mismatch repair. Another remarkable feature of bacterial replication is its rate. The replication of an entire bacterial chromosome in approximately 40 minutes at 37 degrees centigrade requires that each replication fork move about 1000 nucleotides per second, which is equivalent to the length of entire Okazaki fragment. Thus, the entire process of Okazaki fragment synthesis, including formation of an RNA primer, DNA elongation, and stimulation, proofreading by the DNA polymerase, excision of the RNA, its replacement with DNA, and strand ligation occurs within a few seconds. Although it takes E. coli cell approximately 40 minutes to replicate its DNA, a new round of replication can begin before the previous round has been completed. Consequently, When these bacteria are growing at their maximal rate, they double their numbers in about 20 minutes. After initiation, there is second phase or stage, that is elongation. Elongation is undertaken by another complex of proteins. First, replication exists only as a protein complex associated with the particular structure that DNA takes at the replication forks. It does not exist as the independent unit. As the replication moves along DNA, the parental strands unwinds and the daughter strands are synthesized in elongation. Two different but related operations present for DNA elongation. First is the leading strand synthesis and second is the lagging strand synthesis. Several enzymes at replication forks are important to the synthesis of both strands. Parent DNA is first unbound by DNA helicase and the resulting topological stress is relieved by the topoisomerase. Each separate strand is then stabilized by SSB protein. Deoxynucleotide triphosphates are added to this primer by DNA polymerase III. Leading strand synthesis then proceed continuously, keeping pace with the unwinding of the DNA at the replication fork, and RNA primer is synthesized by the primase, and as in leading strand synthesis, DNA polymerase III binds to the RNA primer and adds deoxynucleotide triphosphate. Lagging strand synthesis is accomplished in short Okazaki fragments. DNA synthesis continues until the fragment extends as far as primer of the previously added Okazaki fragment. The new primer is synthesized near the replication fork to begin the process again. See in the diagram, it is clearly mentioned that leading strand synthesized continuously, whereas the lagging strand synthesized discontinuously with the help of different enzymes, that is helicase, primase, gyrase, SSB protein, DNA polymerase first and third. You can see it in the figure, that is in elongation part. So, for elongation, we can say that lagging strand synthesis is continuous synthesis. It begins with the synthesis by primase, that is DNA G protein, of a short about 10 to 60 nucleotides RNA primers at the replication origin. DNA oxyribonucleotides are added to this primers by DNA polymerase III. Leading strand synthesized then proceed continuously, keeping pace with the unwinding of DNA at the replication fork. Lagging strand synthesis, it is accomplished in the short Okazaki fragment. First, an RNA primer is synthesized by the primase, and as in the leading strand synthesis, DNA polymerase III binds to the RNA primers and adds deoxyribonucleotides. On this level, the synthesis of each Okazaki fragment seems straightforward, but the reality is quite complex. The complexity lies in the coordination of the leading and the lagging strand synthesis. Both strands are produced by a single asymmetric DNA polymerase III dimer, which is accomplished by looping the DNA of the lagging strand, bringing together the 2 points of polymerization. The discovery that one strand was synthesized as a small fragment was made by Rishi Okazaki of Nagoya University, Japan, following various types of labeling experiments. The enzyme that joins Okazaki fragments and makes the continuous strand is called DNA ligase. Now in next slide, the entire complex responsible for coordinated DNA synthesis at the replication fork is a replisome. The replisome promotes rapid DNA synthesis, adding 1000 nucleotides per second to each strand, that is a leading or the lagging. Once an Okazaki fragment has been completed, its RNA primer is removed and replaced by DNA polymerase first, and the remaining nick is sealed by DNA ligase. Final step in the synthesis of lagging strand segment, that is RNA primers in the lagging strands, are removed by the 5' to 3' exonuclease activity of DNA polymerase first and replaced with the DNA by the same enzyme. The remaining nick is sealed by the DNA ligase. In elongation, each of core complexes of the holoenzyme synthesize one of the new strand of DNA because the clamp loader is also needed for unloading the beta complex from DNA. The two cores have different abilities to dissociate from DNA. This corresponds to the need of synthesize continuous leading strand where polymerase remain associated with the template and a discontinuous lagging strand where polymerase repeatedly dissociates and reassociates. The clamp loader is associated with the core polymerase that synthesizes the lagging strand and plays a key role in ability to synthesize individual Okazaki fragments. So, proteins at replication fork is SSB protein that bind to the single DNAs, DNA B or helicase protein, primase or DNA G protein, DNA polymerase third, DNA polymerase first, DNA ligase and DNA gyrase. We have already discussed all these proteins in initiation section. Next or last step of DNA replication is termination. At the end of the replicon, joining or termination replications are necessary. Following termination, the duplicate chromosomes must be separated from one another. Eventually, the two replication forks of circular E. coli chromosome meet at terminus region containing multiple copies of 20 base pair sequences called TER or terminus. This TER sequence recognized by TUS protein and makes TER-TUS complex. The TER sequences are positions on the chromosome in two clusters with opposite orientation. The TER sequences are arranged on the chromosome to create a sort of trap that a replication fork can enter but cannot leave. The TER sequences function as binding sites for a protein called TUS, that is the terminus utilizing substance. The TER-TUS complex can arrest a replication fork from only one direction. Only one TUS-TER complex function as per replication cycle. The complex first encountered by either replication fork, they cannot be separated except by the action of topoisomerase. In E. coli, a type second topoisomerase known as DNA topoisomerase 4 plays the primary role in the separation of catenate chromosomes, transiently breaking both DNA strands of one chromosome and allowing the other chromosome to pass through the break. So, when either replication fork encounters a functional TER-TER complex, it halts. The other fork halts when it meets the first or arrested fork. The final few hundred base pairs of DNA between these large protein complexes are then replicated by as an yet unknown mechanism, completing two topological interlinked, that is catenated circular chromosomes. DNA circles linked in this way are known as catenins. Separation of the catenated circles in E. coli requires topoisomerase 4, that is type 2 topoisomerase. The separated chromosomes then segregate into the daughter cells at cell division. The terminal phase of replication of other circular chromosomes, including many of the DNA viruses that infect eukaryotic cell, is similar. Next is regulation of DNA replication. Initiation is the only phase of DNA replication that is known to be regulated, and it is regulated such that replication occurs only once in each cell cycle. The mechanism of regulation is not yet well understood, but genetic and the biochemical studies have provided a few insights. The timing of replication initiation is affected by the DNA methylation and interaction with the bacterial plasma membrane. The ORI-C DNA is methylated by the DAM methylase enzyme, which methylates the N6 position of adenine within the palindromic sequence, that is the 5' GATC. DAM is stand for DNA adenine methylation. The ORI-C region of E. coli is highly enriched in GATC sequence. It has 11 of them in its 245 base pair, whereas the average frequency of GATC is in the E. coli chromosome as a whole is 1 in 256 base pair. Immediately after replication, the DNA is hemimethylated. The parent strands have methylated ORIC sequences, but newly synthesized strands do not. The hemimethylated ORIC sequences are now sequestered for a period by interaction with the plasma membrane. The mechanism is unknown. After a time, ORIC is released from the plasma membrane and it must be fully methylated by DAM methylase before it can again bind DNA A. Regulation of initiation also involves the slow hydrolysis of ATP by the DNA A protein, which cycles the enter between the active with bound ATP and inactive with bound ADP, forms on a time scale of 20 to 40 minutes. This is overall prokaryotic DNA replication. Now, I am going to discuss little bit about eukaryotic DNA replication and its comparison with prokaryotic replication. Eukaryotic DNA replication, origin of replication called autonomously replicating sequences, that is ARS or replicators, have been identified and best studied in yeast. Yeast replicator span approx 150 base pair and contains several essential conserved sequences. About 400 replicators are distributed among the 16 chromosomes in a haploid yeast genome. Initiation of replication in all eukaryotic cells requires a multi-subunit protein, the origin recognition complex, that is ORC, which bind to several sequences within the replicator. ORC interact with its regulated by the number of other proteins involved in control of eukaryotic cell cycle. Two other proteins, that is CDC6, which affecting the cell division cycle, and CDT1 bind to ORC and mediate the loading of heterohexamer of mini chromosome maintenance protein, that is MCM2 to MCM7. The MCM complex is a ring-shaped replicative helicase analogous to the bacterial DNA B helicase. The CDC6 and CDT1 proteins have a role comparable to that of the bacterial DNA C protein. Loading the MCF helicase onto the DNA near the replication origin, two protein complexes have important function in eukaryotic DNA replication. RPA, that is replication protein A, it is a eukaryotic single-stranded DNA binding protein, equivalent in function of E. coli SSB protein. Next, RFC, that is replication factor C, it is clamp loader for pCNA and facilitates the assembly of active replication complex. The subunit of RFC complexes have significant sequences similarity to the subunit of bacterial clamp loading complexes. The termination of replication on linear eukaryotic chromosomes involve the synthesis of a special structure called telomeres at the end of each chromosome. Like bacteria, eukaryotic have different type of DNA polymerase. Some have been linked to the particular function, such as replication of mitochondrial DNA and the nuclear DNA. The replication of nuclear chromosome involves DNA polymerase alpha is association with DNA polymerase delta. DNA polymerase alpha is typically a multi-subunit enzyme with similar structure and properties in all eukaryotic cells. One subunit has a primase activity and the largest subunit contain the polymerization activity. However, this polymerase has no proofreading 3' to 5' exonuclease activity, making it unsuitable for high fidelity DNA replication. DNA polymerase alpha is believed to function only in the synthesis of short primers that containing either RNA or DNA for Okazaki fragments on the lagging strand. These primers are then extended by the multi-subunit DNA polymerase delta. Yet another polymerase, that is DNA polymerase epsilon, replaces DNA polymerase delta in some situation some as in DNA repair. DNA polymerase epsilon may also function at the replication fork, perhaps playing a role analogous to that of the bacterial DNA polymerase first, removing the primers of Okazaki fragments on the lagging strands. DNA polymerase gamma is having polymerization activity and 3' to 5' exonuclease activity and involved in mitochondrial DNA replication. So, as we learn about various types of proteins in prokaryotic replication and the eukaryotic replication, thus there is a need to summarize in the form of comparison between these proteins, which is given in second last table and slide. Here is the name of prokaryotic and the eukaryotic enzymes and their functions. Protein which have same function in prokaryotic replication and the eukaryotic replication having the different names, like DNA A protein in prokaryotes and ORC protein in eukaryotes having same function, that is recognition of origin of replication. In last slide, you can read the difference between prokaryotic and eukaryotic replication. You will get complete idea of this slide after eukaryotic DNA replication. Now in summary, DNA synthesis occurs by semi-discontinuous replication in which the leading strand of DNA growing 5' to 3' is extended continuously, but the lagging strands that grows overall in the opposite 3' to 5' direction is made as short Okazaki fragments, each synthesized 5' to 3'. The leading strand and each Okazaki fragment of lagging strand initiate with an RNA primer that is extended by DNA polymerase. Bacteria and the eukaryotes each possess more than one DNA polymerase activity. DNA polymerase third synthesize both lagging strand and the leading strands in E. coli. Many proteins are required for DNA polymerase third action and several constitute part of the replisome within which it function. DNA B proteins, the helicase activity at a replication fork, this depends on ATP cleavage. DNA B may function by itself in ORIC replicones to provide primesome activity by interacting with DNA G protein, which provides the primase that synthesize RNA. The common mode of origin activation involves an initial limited melting of the double helix, followed by more general unwinding to create single strand. Several proteins act sequentially at the E. coli origin. Replication is initiated at ORIC in E. coli when DNA A binds to a series of 9 base pair repeats. This is followed by binding to a series of 13 base pair repeats, where it uses hydrolysis of ATP to generate the energy to separate the DNA strands. The pre-primary complex of DNA C and the DNA B displaces DNA A. DNA C is released in a reaction that depends on ATP hydrolysis. DNA B is joined by the replicase enzyme and the replication is initiated by two forks that set out in opposite direction. The availability of DNA A at the origin is an important component of the system that determines when replication cycle should initiate. For this lecture, I have referred two reference books. First is Larranger Cox and second is Carr for Molecular Biology. Thank you.